Dear students, very good morning to one another. I am Dr. Nallamma, working as a professor under Department of Pharmaceutical Analysis at SNS College of Pharmacy and Health Sciences at Coimbatore. In this B Form first sem, under this pharmaceutical analysis one theory, under this first unit we have errors topic. In this previous video we was discussed about the sources of errors. Under the sources of errors, today we are going to discuss about method errors and environmental errors. We are going to discuss. What do you mean by method errors? Method means here it will apply for the procedure. So procedure. So if you want to do any pharmaceutical formulations, like maybe it's a parenteral or a tablet or a capsules or a transdermal batch or any other cosmetics, after the manufacturing or during manufacturing or selection of raw materials, we will perform this quality control test actually. In this method errors, so if you are taken for the tablet, okay, paracetamol as a solid dosage forms, in this lot number 301, you need to find out this percentage purity or amount present. So we need to follow IP Indian Pharmacopoeia. The Indian Pharmacopoeia was given for okay which solvent okay we need to use. So how to take this average weight of the tablet and from that average weight of the tablet, the required weight okay we need to calculate. So these are the calculations or these are the formulas clearly they given in this Indian Pharmacopoeia. Or you can take any monograph, okay, or ICH guidelines we need to follow. So these monographs has any substandard, okay, such methods we cannot use. So if you are using any substandard procedures or methods, that is called as method errors. Incorrect or unsuitable analytical method if you are selecting, we won't get proper result. So if you are selected for the wrong procedure or wrong monograph or wrong guidelines, then we won't get proper result. So we cannot conclude the quality of the product with the help of such wrong procedure. That is the reason we need to select the standard protocols like IP Indian Pharmacopoeia or British Pharmacopoeia or United States Pharmacopoeia we can use. Even European Pharmacopoeia also available. So any one standard pharmacopoeia procedure we need to perform. We need to arrange all the solvent systems and the needed instrument, and we need to select okay, proper lambda max and everything. Finally, we can find out the absorbency. From the absorbency, we can calculate the concentration. Through concentration, we can find out amount present. So, if you are selected the wrong method, such errors is called as method errors. This is the third types of sources of errors. If I am moving to the fourth type, environmental errors like temperature or humidity or light affecting result. So at the industry we have HVAC system. We need to provide proper temperature and humidity and light and airflow also, and we need to avoid contamination, particularly these bioanalysis or bioanalytical activities when we are performing. So the freezing point or storage okay, temperature very important. Even vaccine manufacturing unit also the temperatures play vital role. So if you are provided for the wrong temperature or a wrong humidity or more light or less light, it will affect the data. So these are all will come under environmental errors. The next one, the types of errors. The major three types we have: determinate, systemic errors, random errors, the third one, major mistakes. So what do you mean is systemic errors? Consist and reproducible. So these systemic errors can be reproducible and it can be correct. We can correct it. So such errors called as the systemic errors. The second type was random errors occurs by chance, cannot be predicted. So such random errors cannot be predicted easily. Then the major mistakes such as recording wrong data or calculation errors, this is called as major mistakes. This can be predicted easily and we can correct it easily. So once again I am repeating the types of errors. We have three major types. One is systemic errors, second one is random errors, the third one major mistakes. Thank you all. I hope this session was very useful for you. The further discussion related to errors we will see in the next session. Thank you all. Have a nice day.